won't spend quite as much time on these, but give a bit of an overview of where we are, because these invasive pests are also of interest. One thing I wanted to mention about a lot of these invasive species programs or research programs in North America is that we have a National Institute of Food and Agriculture grant program focused on what we call specialty crops, and the specialty crop includes tree fruit crops and small fruits and things like that that aren't grown in huge acreages of field crops. And a lot of the research that's been done in the last couple of decades on brown marmorated stink bug was also done with two of these big specialty crop research initiatives. And these bring together researchers in a wide range of institutions to all work cooperatively. So it's a real good example of a cooperative research program among people in different institutions. So we also have just finished, are just finishing up a specialty crop research initiative grant on spotted wing Drosophila management. And some of the research on spotted wing Drosophila has also focused on methods of detecting it, attracting it for mass trapping, attracting it to detect it, attracting it to to help determine when spraying needs to be done or when management needs to be done. And a lot of different methods have been experimented with as lures for spotted wing Drosophila. So there's a commercial pheromone lure. I guess the battery's dying on the pointer, which is commercialized and available from Trace, and I think there's a second commercial lure available from another country, pheromone lure. There are also various other types of chemical attractants. Some of them are very simple. Spotted wing Drosophila responds very easily to simple vinegar lures, vinegar and water, and this is something that is often promoted for use by farmers because they can just mix them up and put them out in the field themselves. The disadvantage of a lure like that is it attracts lots of other stuff too. So it means sometimes it means lots of sorting is needed, but it is an effective trap and it's simple to use. Other products have been developed that are a bit more specific, like the Suzuki trap and the Drosa lure, and so on. So a lot of work has been done developing compounds like this. Some of the more recent interesting work has been done with erythritol, which is found to be an attractant and also toxic to spotted wing Drosophila. Erythritol is the active ingredient or a component of active ingredient in one of these artificial sweeteners. I forget the brand name. Maybe somebody knows. So something that people are using to sweeten their coffee and tea is toxic to spotted wing Drosophila. Kind of interesting. So of course, with any new invasive pest, a lot of work is done on testing chemicals for their efficacy against the invasive, and that's certainly been the case with spotted wing Drosophila. Growers, again, with the advent of a new invasive species, usually have to fall back on the use of pesticides to manage the crop until research identifies other means. And this is just an example of a wide range of chemicals, of pesticides that were tested against spotted wing Drosophila, and you can see the dotted line indicates where what's considered a good level of control, and many of the chemicals don't meet that. There are a range of chemicals that are available that do provide reasonably good levels of control that growers can use. However, the one interest is spinosad. So this one is available for a wider range of crops because it can be used. Theoretically, it's less toxic than most of the other pesticides, and it provides reasonable levels of control. Just two examples of testing regimes and chemicals used in different parts of the U.S. where blueberries are grown in large number. Michigan and the north central states, and Georgia in the southeastern U.S. And from the blueberry, the Michigan blueberry spray program on the left, you can see that even though the chemicals are effective, they have to be used regularly. So they have a spray program, week 1, 2, 3, 4, 5, 6, 7, 8. That's a lot of sprays. And it had been even worse originally, so they've been able to get the number of sprays down, but that's still a lot of sprays, a lot of pesticides being put on, a lot of additional cost and effort on the part of the growers. From the diagram on the Georgia blueberries, you can see they've tested and recommended a number of chemicals that work pretty well, or work under short pre-harvest interval conditions. Which need to be used right shortly before harvest. There are fewer chemicals that can be used, for example. Fewer still chemicals that can be used in reduced risk or organic crop situations. But there are some recommendations. So particularly for organic growers and reduced risk, spotted wing Drosophila remains especially hard to control because they have fewer tools that they can use. And there's also some work that's being done in genetic methods of controlling spotted wing Drosophila. I'm not so up on that, and much of that's not published yet or available or publicly available, so I can't say much about that. But I will say a few words about our research in biocontrol for Drosophila suzukii in the U.S. And a lot of this work has been done in conjunction with colleagues on the West Coast, the University of California and Oregon State University. West Coast is where spotted wing Drosophila was first discovered in North America in around 2000. And within the space of 2 or 3 years, spotted wing Drosophila was everywhere across the U.S. So it spread very, very rapidly, much more rapidly than brown marmorated stink bug. So again, one of the first things we do in a program of research against invasives for biocontrol is try to determine what the native species are that may be attacking new invasive, spotted wing Drosophila in this case. And of course, Drosophila as a group is an insect, a fly about which lots is known because it's a common lab rat insect. A lot of work has been done on behavior of parasitic Hymenoptera using Drosophila species and Drosophila parasitoids. So we know quite a bit about Drosophila parasitoids in general. In North America, it turns out that there are some species of resident larval parasitoids, which typically are known to attack Drosophila species. And the big genera of larval parasitoids are Asabara and Leptopolina, and these are braconid, tiny, tiny little braconid parasitoids that attack the young larval stage and then emerge from the pupa. And there are also 2 species of resident pupal parasitoids in North America. And these pupal parasitoids occur worldwide in the northern hemisphere, so they're not unique to North America. The pupal parasitoids are not specialists at all. They attack a very, very wide range of flies. They can be easily reared, but in nature they attack spotted wing Drosophila at very, very low levels, just a couple of %. The resident larval parasitoids, and some larval parasitoids are very effective parasitoids of their drosophilid hosts, but the ones that we have in North America and in Europe that attack other Drosophila species are not capable of developing in spotted wing Drosophila to a large degree. Very much like the situation with the native Trisulcus, which don't develop well in the invasive brown marmorated stink bug. These larval fly parasites don't develop in spotted wing Drosophila because spotted wing Drosophila have physiological defenses against these parasitoids that usually will kill the egg or kill the young larvae, preventing it from developing to adult. So again, we look to Asia for adapted parasitoids specifically of spotted wing Drosophila, and we found a pretty good variety of parasites that do attack spotted wing in the native range, which again is China, Korea, and Japan. A fair number of braconids, all in this genus of Asabara, several species of fidgeted wasps, Leptopolina species, again, and species in the genus Ganaspis. And also in Asia, these same 2 pupal parasites that we have in North America and in Europe. In Asia, the situation is a bit complicated because spotted wing Drosophila and Drosophila suzukii often co-occurs in the same area in the same fruit with two other species that are very closely related, this Drosophila subpulchrella and Drosophila pulchrella. They're difficult, they're not easy to tell apart, and they both occur in fresh fruit. All three of them can co-occur in fresh fruit. But the parasites seem to attack all three of these. So in North America, we only have Drosophila suzukii, but if we were to get the other two species, the parasites that we've collected from Drosophila suzukii, probably some of them would attack the other two species as well. So this is a list of the species from Asia, from spotted wing Drosophila, and the three species in red are the most abundant. They seem to be the most important. Some of these other ones are incidental, collected only in very small numbers. And when we collect them from fresh fruit, these are the species that rear out. Ganaspis brasiliensis is the one in brownish orange. Leptopolina japonica is the pale green, and Asobara is in blue. So you can see that the two phagitids are the dominant parasitoids in South Korea and in China. And these are the species that we pinned our hopes on for classical biological control. Now, in the host range tests that were done in California, which included these two pupil parasites, you can see the range of non-target test species on the left side represents about 25 species of Drosophila and other genera that are close to Drosophila. Oops. And the bars under Trichopria and Pachycropoides show their reproduction on each of these non-target hosts. And you can see that both of the pupil parasitoids are pretty much generalist. You know, they attack most of those species to varying degrees. Some of them, the Pachycropoides, to a pretty high extent. That has implications for potential augmentative biocontrol because both of these species are easy to rear. And because they're cosmopolitan in the northern hemisphere at least, they can be reared and released in different locations. And there are researchers in the US and in Europe who are experimenting or interested in using these augmentatively. These are the results of the host range tests of the larval parasites, which in order to introduce them, we have to demonstrate some host specificity. And you can see that Asobara japonica, the braconid, is not very host specific. And we would never get permission to release that in North America. But the two phagitids, Leptopolina and Ganaspis, are specific enough. And a petition was prepared for both species, submitted, returned with a request for further information, and then resubmitted specifically for Ganaspis brasiliensis. And it's pending, and we're hopeful that it will come back with a positive recommendation for release. This is the Ganaspis brasiliensis. One of the two most specific parasites. Maybe not the most active on spotted wing Drosophila, but the parasitism levels we see in Asia are high enough, 30, 40, 50, 60, as high as 70% in some habitats. We think it's high enough to have an impact on spotted wing Drosophila in reservoir populations particularly. And spotted wing is like the MSB because of its broad range of hosts. Biocontrol in unmanaged reservoir populations is really the only technique we can rely on to help bring those populations down. So in summary, for spotted wing Drosophila, we've learned a lot about their biology and ecology in recent years, quite a bit about developing effective baits and lures that are now available commercially. Learned quite a bit about insecticides that work and are available. One interesting point about their biology and ecology is that the spotted wing is most active as an adult fly right at dawn and dusk. And the recommendations are to make their insecticide applications during that period of the day, which is when most of the adult flies are going to be in contact with the pesticide. Further work needs to be done for tools to manage the flies in organic situations. A lot of growers are using completely screened enclosures to keep flies out. That works. It's more expensive, but it works. And that's a situation where augmentative biocontrol with these pupal parasites could be useful because it would be a semi-contained environment. Let's see, and I think next I'll move on to invasive spotted lanternfly. A lot of spotteds coming into North America. Some of you may have seen photographs of this insect. The adults are about this big. It's a good-sized insect, very easily recognized. They do occur in tremendous numbers in the field, and the picture on the upper right-hand corner is an apple showing the large numbers of adults which aggregate on host plants at the end of the season when they're starting to find places to lay their eggs. The picture in the middle and the bottom shows large numbers of adults on grape. They really do get to be that thick on tree trunks. They have the lanternfly has one generation a year, which makes it much nicer to work with than drosophila, for example. The eggs hatch usually in the spring, and they go through 4 nymphal stages. The adults show up in midsummer in June, and then feed for several months, and then they start to aggregate on certain host plants to lay their eggs in the early fall, at the end of September and October. And then once they lay their eggs, that's it. The females die shortly thereafter. The males die. Only the eggs survive the winter. Again, we have just gotten a specialty crop research initiative grant to start doing research on a whole aspect of lanternfly biology and management, including people from many different institutions. This shows where lanternfly is in the East Coast, and this is the part of the East Coast that referred to as the Mid-Atlantic states. So this is Delaware. Here's Washington, D.C. Here's New York, Pennsylvania, Virginia. BMSB came into the U.S. right about here. Lanternfly came into the U.S. in almost exactly the same spot. It may have followed a similar invasion pathway by coming in on imported materials. And in fact, it's thought that lanternfly arrived on rock that was quarried in China and shipped over, because the lanternflies will lay their eggs on many different surfaces. Modeling has been done with Climax to show where lanternfly is likely to become established in other parts of the world, and you can't quite see too much detail here for New Zealand, but there are colors on here that suggest that it could survive here. The climate would be suitable, and you can see large areas of Australia and temperate parts of North America and Europe are included in parts of South America as well. And so there's no reason to think that it might not be capable of surviving over a fairly wide range of introduced zones. And I mentioned the egg deposition. The eggs are laid in clusters, kind of like stink bug eggs. They don't look like stink bug eggs, but they're laid in clusters up to an inch or so in size, and then they're covered with a foamy material that dries and changes color to look basically like mud. Yeah. And they can be laid on in any kind of a surface, a light bulb, lawn furniture, old tires, metal fences, telephone poles. In nature, most of them are laid on the trunks and branches of trees at some a couple meters off the ground and higher. But they also will lay them on rocks pretty readily. So you have searching for them, you have to look at a wide range of surfaces in order to be able to pick them up. The crop of particular concern for spotted lanternfly is grape. To some extent, other tree fruit crops like apple. You saw from that earlier picture that tremendous numbers would show up and feed on apple at the end of the season. And even though the lanternflies are sapsuckers, phloem feeders, they're not feeding directly on the fruits themselves, but they're feeding on phloem from the trunk and the branches. And they're producing tremendous quantities of honeydew, which contaminates the fruit. And they can feed on large enough amounts of phloem that they're weakening the plant. And with apples, the concern was that they would damage the buds that would be producing fruit the following year. So far, there hasn't been evidence of obvious impact on apples, but there is definitely evidence of significant impact on grape. Spotted lanternflies do have a rather wide host range. It's not as wide as an invasive like brown marmorated stink bug or spotted wing drosophila, but they feed on quite a range of trees and shrubs in a wide variety of different plant families, so they're not restricted taxonomically to a certain group of hosts. And this just shows a range of hosts: tree of heaven, grapes, walnuts, maple trees, willows, sumac. In Asia, some of their favorite hosts are tree of heaven, Ailanthus. I don't know if that occurs here. Is that an invasive? Chinaberry they feed on. There's another Asian tree called Chinese tuna, which is grown widely in Asia. They really like that. They do quite well on sumacs. In Pennsylvania, they've been shown to feed on walnut to the extent that the walnut trees are stressed and damaged. And walnut's of concern in the U.S. because it's an important timber species as well as the nut producer. There's had been some thought early on that they had such an obvious affinity and a liking for Ailanthus that maybe Ailanthus is a species that's required for its development to producing eggs all the way through adulthood and egg production. Well, that turns out not to be the case. They can't develop all the way through to adulthood and reproduce laying eggs on other host plants besides Ailanthus, even though they definitely like Ailanthus a lot if it's around. And they seem to have a wider host range in the younger nymphal stages than they do as an adult. And as they get older and they molt and they turn into adults, they seem to go to fewer and fewer hosts. But the host that they will feed on at the end of the season as adults still is a pretty wide range. It includes apple and grape and maple trees, a lot of common ornamentals. They're starting to spread, and the bigger the distribution gets, naturally, the easier it becomes for them to be moved around as a hitchhiker, because their populations are just growing and growing and growing. And at the end of the season, when they're moving around in large numbers, they'll fly into cars, vehicles, railroad cars, and they're shipped around the country. They've been found in airplane bays. They can move around pretty well on their own too. The nymphs can very easily move from plant to plant, from tree to tree. They're active jumpers. They can jump several meters. They're very easily disturbed. So if you come up and you try to collect them, they're likely to jump away. They have this habit of climbing up and down trees during the day. So they move down the tree at night and they move back up again during the day to feed on the branches. It shows some of the older stages of nymph. And some of the hosts on the left that they can complete their development on. Again, tree of heaven, walnut, chinaberry, certain oak species. You would never really guess which plants they can utilize effectively because it covers such a broad range. Hops is another plant of commercial interest. So both wine and beer are at threat from lanternfly. And I mentioned that damage has been shown in grapes. Grape growers are starting to use extensive pesticide applications. They have reported significant yield loss and vine death from lanternfly. The average number of insecticide applications on grape went from 4 before lanternfly to 14 in just 2 years. So that's that's like tripling the cost of insecticide applications. That's a big extra cost. This is an example of one of the first orchards that was impacted by lanternfly in Pennsylvania. This is after 2 years of lanternfly feeding. And this grower basically has lost all of his production, and even the vines, if they're not killed, the heavy feeding prevents them from producing the fruit, and some varieties like Riesling have been killed outright. So this grower has basically abandoned his vine production and is moving to something else. This is another shot of a grapevine in September. These plants should be full of leaves and full of fruit at this time, and you can see that they're not. Management at this point is still rather basic. For several years, APHIS in the state of Pennsylvania was actively trying to eradicate it. Every year they tried harder to eradicate it, and every year it continues to spread. So eradication is pretty much off the table now, but there are still efforts being made to slow the spread. People are encouraged to scrape the eggs where they find them, but again, in trees, most egg masses are 2 meters and above, where they can't be seen or can't be reached. So that's not really a very effective management tool. Basically, it's just a feel-good measure. Trees can be banded, putting sticky bands around the trunk, and that will trap large numbers of nymphs and adults as they walk up and down. Because they like to feed on tree of heavens are recommended for removal, but again, it's such a widespread weedy species in North America that nobody will ever get rid of it. It's just too widespread to remove entirely. But it has been useful as to be used as a trap tree. So certain trees that appear to be more or especially attractive to the lanternfly have been treated with neonicotinoid or surface chemicals like ditefluron, and then the adults that go to that tree are killed. This is an example of banding and showing the large number of nymphs and adults that get caught up in these traps. But again, that's more of a monitoring tool than a removal tool. You can never hope to remove all the lanternfly in an area with just by using a banding program. Fortunately, lanternfly are pretty easy to kill with the pesticides that are available, and you can see the numbers on these grapevines that have been killed. It just gives you an idea of the number that occur. It's really quite astonishing. There's a lot of work trying to develop attractants and pheromones. Nobody has discovered a chemical attractant or a pheromone yet, but they do have, because of their behavior of crawling up and down, they will walk into physical traps. And this is the best method for trapping and monitoring that people have discovered so far. We've been monitoring for native natural enemies in North America, and so far we haven't found anything that looks too promising. There are general predators that will attack, that will try to capture things that they're capable of trapping and eating. So we have some predatory insects, penatomids and larger rediviids, spiders and praying mantises and so on that have been seen capturing and eating lanternflies, but not in great enough numbers to have any impact at all. We have been monitoring for parasites of the eggs and the nymphs. Have not found anything in the nymphs yet. There has been recovery of small numbers of a parasite, not abundant enough to have an impact, but this is an interesting parasite, Oencertus cuvanae, because it's an exotic parasite itself, and it was introduced against gypsy moth about 100 years ago into North America. And we have not found this parasite in lanternfly in Asia, which is where this parasite comes from. So it's kind of an interesting twist. I suspect that it probably is a spillover onto lanternfly from areas where gypsy moth populations are high, which happens from time to time. Several fungal pathogens have been found. in Pennsylvania. One of them is, I believe it's a native species, Batcoa major, found on significant numbers of adults in a couple of locations at pretty high levels, 80% mortality. And the other species is the Bavaria bassiana strain, found in the same year in 2018. And this was a year when it was exceptionally wet, much wetter than usual, which probably contributed to the outbreaks of these pathogens because they haven't been seen regularly in other years. But Bavaria bassiana can be used as a bioinsecticide, so there are trials going on to see if Bavaria strains can be applied against existing populations of lanternfly. So that could be a possible means of management. Works against the adults and the nymphs. So in classical biocontrol possibilities, we've started surveys in Asia, and there appear to be two, at least, dominant parasites. There's one egg parasite in the genus Anastatus. A lot of Anastatus species also attack stink bugs. And there's one nymphal parasite in the genus Dryaenus. These are very interesting insects. You may not be able to tell here, but Dryaenus species have raptorial tarsi. So kind of like a mini praying mantis leg at the end of their front tarsi, and they use those to grab the host and immobilize them while they parasitize them. Another unique thing about the Dryaenus is that as the larvae start to grow, they start to extrude from the body of the nymph, and they create this sac called a thylacium, this dark thing. It looks kind of like a round aphid mummy. And when the larvae gets bigger, most of its body moves into that thylacium, and the head stays inside the host and feeds inside the body of the nymph. And then when they finish feeding, they completely drop out of the host, and they spin a cocoon on tree bark or wherever, and then they spend the rest of the year in the cocoon stage and reemerge the following spring. So at this point, we're still doing biological studies on these two parasites. We haven't figured out yet how to rear the Dryaenus. The Anastatus is easy to rear, so we're further along on that. We've surveyed good parts of Asia where Lycorma occurs. The red and the yellow are the parts of China where Lycorma is easiest to find, and we've looked in a good number of those areas. There's still a possibility, we think, that there could be some cryptic parasitoids that we haven't discovered yet, and we're not finished with our surveys. But we've moved on to host range testing in North America. Unlike brown marmorated stink bug, where there are almost 400 species of native or resident stink bugs in North America, there are only 19 species of fulgurids in North America. Most of them occur in the southern part of the U.S. Rather than where Lycorma occurs in Pennsylvania. So we've had to stretch our thinking a little bit to find suitable host range non-targets to test, but there are other plant hoppers that have similar biologies that lay their eggs in egg masses and above ground that we're putting into the testing scheme. Many of them have never been studied before, so we don't know where they lay their eggs. We don't know how to rear them. So that's one of the difficulties in starting this kind of a testing scheme. We're also testing the egg parasitoid against other stink bugs that we know other Anastatus species can attack, which include other hemipterans like stink bugs and squash bugs, rediviids, and also various lepidopterans. And these are just some preliminary results. They look very much like our stink bug results. Many of the non-targets are not attacked, but some of them do have low levels of attack in groups that would be of concern for non-targets. So, for example, the non-target in the green bar on the left-hand side is another leafhopper, but the one on the right-hand side here, NASA, is a squash bug. It's a pest, but it's a completely different group of insects. And again, the parasites that rear out of these non-targets often are smaller and potentially less fit. This is the Publicia is the species of leafhopper or plant hopper that's most similar to Lycorma in North America, showing the egg masses of the two different species. We've done some chemical ecology work with Anastatis, the egg parasitoid similar to what I described for Drosophila japonicus and brown marmorated stink bug. And these egg parasites are capable of detecting the pheromone that Lycorma leaves on the tree surface. And further, we've also compared their response to Lycorma and the Publicia, the other leafhopper or plant hopper. And as with Drosophila japonicus, they respond much more strongly to the pheromone of Lycorma, the spotted lanternfly, than they do to the other. So again, it's always an encouraging sign with regard to potential host specificity under field conditions. But we're not nearly as far along in this research as we are for brown marmorated stink bug. So a lot more to be done with spotted lanternfly. We're just getting started. And that's what I have.
Thank you very much to Tim and Elijah.